It's a pleasure to be here and to see old friends and meet new families. It's an honor for me to speak to the group. I know many of the families here for many years. And I'm going to talk today about the approach that we've taken over the years to try to help develop new treatments. It's going to focus more on gene therapy just because I can't go over everything in the short time. And just to be warned up front, it's going to be, some of it's going to get highly technical and then some of it won't be as technical. So for the technical stuff, if you want to stop me or get me afterwards for the explanations, I would appreciate that. I'm here with a number of people in the group that could also help. So let me just get into this. This is sort of like going to be a story today with 3 chapters in it. One is going to be the background. It's a little centric to me and a little bit about organic acidemia research. The next thing I was going to comment on about is for the group is about models and biomarkers. We've been hearing a lot of people talk about biomarkers. And I want to just try to make people understand how important that is and how difficult of a problem it is for some of the disorders that the children and young adults have. And then talk about new genomic therapies, and that's a group of therapies. I'm going to focus on AAV gene therapy, which is something we've been working on for a long time. So let's get ready for part 1. I have some disclosures, and you can see I have a number of colleagues that have helped me in the corporate side, and also when we have patents at NIH and they say that the NIH holds the patents. We don't have them. We're not allowed to be in companies, but the NIH holds that, and that whatever happens, the NIH gets, if it licenses, that money goes back to NIH to support research. So what are the inborn errors of propionyl-CoA metabolism? I'm going to group these in a big group, and it's been covered by the number of the speakers, so I'm going to go through this quickly. These disorders are not new at all. They've been around probably since the beginning of human time. They were really first recognized in the 1960s by clinical phenotypes. And the patients with MMA and PA and cobalamin disorders have absolutely massive elevations of these organic acids, MMA and PA. And there's other ones, but those are the ones people recognize. Most of the children in the early literature had either methyl-mannose-CoA mutase deficiency, MMUT, or had propionyl-CoA carboxylase deficiency. And that comes in 2 types, the alpha type and the beta type, and they're pretty much identical. If a patient doesn't have any activity of one, it equals the no activity of the other. It's been long recognized from the early description of these conditions that these are very difficult conditions to treat, and the prognosis is guarded for patients that have the severe forms of these disorders. And they represent a larger group of organic acidemias. Other conditions besides MMA and PA can have similar clinical phenotypes. They don't have the same biochemical abnormalities, but they're very representative of a large group of many, many disorders. And that makes it very important to figure it out and study these. It will apply to all of them. That's the rationale, okay? So it gets complicated quickly. And so it's going to go up a bit here as we go through biochemical pathways. And so if you can see my arrow. Okay, do I have a pointer up here? Let me see this. Give me a second here to find the pointer. Turn this on. Okay, doesn't work. All right, use your imagination. It's a big chart over here, and there's lots of little pathways and bubbles. Anytime there's a problem with one of those, a person can get methylmalonic acidemia, propionic acidemia, hyperhomocystinemia, or a combined form of MMA and homocysteine elevations. And the ones that are most common have a little box around them. PCC A and B deficiency, that's propionic acidemia. MMUT, that's mutase deficiency of one type of isolated MMA. And then cobalamin C deficiency, which is the most common form of the combined problems that produce MMA and high homocysteine. And by the way, if anyone wants to look this up, this is all. You can look up Jen's paper, Jen Sloan. It's online. You can go and read this pathway all night if you want. It's free. All you need is internet connection. Okay. So in the early patients were recognized to have this condition that they called ketotic hyperglycemia. And we just figured out the actual mechanism for this. It took us 40 years as a field. And one of my postdoctoral fellows figured this out, why it happens. There's a very specific reason it happens, which I'm not going to really get into, but if someone really wants to get into this, I'll talk to you afterwards about what the exact mechanism of why the glycine's high in our children. Early investigators noticed these clinical features that are still present today in the patients, that they have intermittent life-threatening episodes of ketoacidosis. Some people might say all of a sudden the children are air hungry. And all of a sudden it's very quick and they get sick so quickly. That was part of the original description. It happens to the patients today still. Many of the patients in the early literature had growth and developmental problems. This is an area of debate and discussion. It is not uniform to have developmental problems in the patients. And we spent a lot of time studying this. These conditions affect multiple organs. You heard about this earlier today. And that's the reason for that is not fully understood. This is why you need research. And there's a complicated intermediate metabolic defects. There's perturbations of other pathways. Again, this makes it hard to figure out what's causing what. And there's a propensity for these patients towards infections, movement disorders, kidney disease, especially in MMA. We know that happens in PA as well. And early authors concluded this, that the conditions were difficult to manage with disease progression and death despite biochemical response to a precursor restricted diet. Early on, it was known you can't just measure the metabolites. That doesn't tell you how well the patient's going to do clinically. And this is from the 1960s and 70s, so don't get too alarmed for the families. But this is part of what you do when you study. You study what people saw before. And that's what people wrote. Now, fast forward to around 2005, and some people in the audience, and I was delighted to see one of my patients from Philadelphia from many years ago, when the state of Pennsylvania was one of the first and really an early leader in the world to institute expanded newborn screening, which eventually became uniform. And in 2005, there were recommendations made. And now all babies are screened in the US for forms of propionic acidemia, methylmalonic acidemia, and cobalamin disorders. And you can see the list here, and it keeps growing. And it really does grow. And these disorders serve as an important paradigm for the next steps of newborn screening. So there's lysosomal disorders, there's immune deficiency syndromes. It's growing and growing. And it won't be far away until we're using metabolites plus genome sequencing on babies. It's already happening at a pilot level, but I think if someone told me this in 1999, I would not believe it. But here I am telling you this is happening, and it will happen. To more of an extent than just studies, right now, pilot studies. And that brings up the question of what are we going to do for all these other infants and children we diagnose with these disorders? We're going to have to develop treatments for them if you screen for them, right? That's part of newborn screening, the mentality of why you screen. You don't screen to just find someone and say, we don't know how to treat this. You should have an intervention. And this is, again, a debated area of policy. Now, this comes up a lot. I heard it discussed earlier. How common are the conditions? And when you have a rare condition, you need millions and millions and millions to have statistical confidence of your number to say it's 1 in a million, it's 1 in 100,000. And this is a very large study from 4.5 million, and it's still probably not enough to really understand what the incidence is. And what you can see here is these are patients that babies were born in the state of California. They received supplemental expanded newborn screening and exome sequencing, so sequencing all the genes to see if you had a metabolite elevation, did you have a mutation in the gene? And this is the results, a very important study published in Nature Medicine. And you can see from this, those are all the conditions. And this is a real estimate of 4.5 million over a 10-year period. This is a pretty good estimate. And this is the numbers. Medium chain acyl-CoA dehydrogenase deficiency, for those who know me a long time, I started off studying that many years ago for this reason. I felt it was very common. We had to screen for this. And I worked with Dr. Berry when I was a fellow to work on this as well. PKU, which is an amino acid disorder. And then the next most common group is MUD MMA. And if you look at them as a group and group them together, the MMA and PA metabolic disorders are very, very common in this group of patients. Now, overall, they're still very rare. But this is an answer to how common is it. So I mentioned the importance of studying this, why this is a sort of a paradigm for the conditions that many of the kids in the audience here have of screening, which is why if you're going to screen, you need a treatment. There's a fundamental debate about this. It's been going on since the 1950s and 60s when people started screening for PKU. And the story really has evolved to this. And I'm showing you this because I think this is really important. This is a disorder called spinal muscular atrophy type 1 that this is talking about, which decades ago was a absolutely uniformly lethal disorder by the age of 2. If a baby had this, 100% chance they're going to die by the age of 2. Over the years, we've and with enough research, now we can diagnose patients. There's even a new gene therapy for this. So now soon every baby born in the US will be screened for this. So this is the paradigm. But we're not there yet with MMA and PA and cobalamin disorders. Yet we're screening for them, you see? So there's a fundamental importance of what we're all doing here today. So I'm going to tell you a bit about me and how I got interested in MMA and the background and why it's important to work in this area and train people. So this is where I did my internship and residency. Massachusetts General Hospital, that's 1911. Okay, I wasn't born in 1911, but that's what it looked like. That's the Bullfinch building for Dr. Berry, the professor at Harvard. That's the Bullfinch building. Patients used to come in by boat. That's all. There's no grass there anymore. But anyways, let's see. I'm not in the photo. So I wasn't even, a thought of anyone's mind yet. So I trained there and I finished up my training and then I moved on to a really great place, which is the Children's Hospital of Philadelphia, around 1999. And at that time, this was happening, newborn screening. And it wasn't mandated by the government yet. It was being done by the state because the state had a pilot program, and there's more to say about it. But we started seeing families that were having positive newborn screens for MMA, PA, cobalamin disorders. That's really where it began. That, and I had some great support from the families early on. So I got interested in MMA. I thought this was a really important problem because we're diagnosing a lot of patients. We don't have a treatment. So I tried to find people that would support this. I found Dr. Berry, who's been a supporter. And then I actually also ended up working with Professor Haig Kazazian, who was the chair of the genetics department, because most people told me, you can't treat MMA and PA, don't work on this, work on something else. Until I met with a few people and I said, no, this is an important thing. You should work on this, especially Professor Kazazian, who gave me space in the lab to start working on MMA in around 2000. The years clicked by and I had an opportunity to move. So a tremendous opportunity to go to the NIH. And it's been amazing. So for those who don't know, right down the street is the NIH. It's the world's largest clinical research hospital. It dwarfs anything else anywhere in the world. We have 300-something research beds. Everybody at NIH is on a research protocol. And we're doing research on MMA and PA. And this is something else. If you are paying taxes in the United States, you have access to this. You can be in a study, and it's free. So if you're interested, we're welcome to see you. You're supporting us, and we're very grateful for the support from the federal government because that's who's funding us. But this is our research hospital. To go back. So I joined right out of fellowship. I left my fellowship and joined the faculty of the National Human Genome Research Institute with sort of a grand idea that we're going to develop this program to study MMA, PA, and cobalamin disorders. But But what I realized back then, and it's still super important, is that clinical research is absolutely critical. And it's very difficult to do this for rare disorders for the reasons you can imagine, but even beyond that with conditions where the children are very, very sick and can have metabolic instability. And I know that some patients have had this happen to them at NIH because it's part of the condition. So it's very dangerous for clinical research from a risk perspective. And in the beginning when I moved to NIH, it's still something we are concerned about because of safety is the number one concern for our families. But then if we could do this in parallel with developing the animal models, then we would have a chance of really getting the insights from animals and patients to try to figure out what are the disease mechanisms, what's causing it, and then to test and develop new therapies, you see? You need both of them, and you need a hybrid approach. And that's great, but who's actually going to do it? Well, we started off the unit in 2004. That was a team. That was Randy, he's my longtime colleague from UPenn. Came down, he was a PhD candidate at the time. And he was really running and leading the laboratory efforts. And there was Jen Sloan, I don't know if Jen is here, I think she's here somewhere. And so we would alternate every other week. Jen was actually seeing patients for another protocol, and we would see patients one week and in the lab the next week. So the team really grew. And then I'm proud to show this picture because as we grew, we were actually also able to attract and retain and get people really interested in MMA, including people that are even I haven't seen in a while. Dr. Carrillo Carrasco is one of my fellows. years ago. There she is. Dr. Hauser is the chief of genetics now at Inova Fairfax. And you can see the names there. And of course, the famous Dr. Manoli, who is really the world's expert on MMA. And then there was other people that weren't pictured. So we continued to get traction and interest, and the group grew. And there's some students over the summertime, just learning about MMA and PA. And then this summer was a good photo because all the students' names there, they all got either MD scholarships or MD PhD scholarships to go to med school at places like Yale and Mount Sinai. Really a wonderful group. Actually, Sam is up in the front, is actually a PhD student at Harvard, and Madeline is a PhD student at Berkeley. So all these people are studying MMA and PA to get ready to get launched into their careers. So it's really been wonderful with that. That's our last picture before COVID struck. So you can see the different people in the group. And then it took almost 2.5 years to take another group photo. And there's some new members, including some of my students that are here with us. And I can't mouse over this, but for those who got the little fluffies and stuffed animals, Pam Head, who's a postdoctoral fellow, and Pauline handmade all those over months and months to get for the children. So that's the group now. Okay. So I did want to thank everybody for their support over the years. And there's a couple of pictures here of everybody at NIH over the years. Lots of our participants. Sort of cool. Okay. So that's how we got to where we're at. I'm going to just quickly go through some of the things we discovered. And I can't go through all of them. Each one of these would be a separate topic. Many things, okay? About metabolism in worms, mitochondrial dysfunction in MMA, kidney disease, side effects of gene therapy, fundamental important studies on medical foods conducted by Dr. Manoli and Dr. Shalachko. And all this time, we were very grateful that the families would be in our study, and we've seen about 262 patients with MMA and about more than 40 with PA over the years. So we have other things we've been doing. We're actually starting to work with zebrafish models. We were working with another institute at NIH to develop a funding model that could help universalize gene therapy for lots of disorders, including not just MMA and PA, but this is called the PAVE-GT program, which I'll tell you a little bit about. We've continued to be very interested in hydroxycobalamin dose escalation as a treatment for cobalamin C deficiency, and developing biomarkers and new gene therapy technologies to treat MMA. It continues. We've been involved with gene discovery for other things. And then I won't have a lot of time to talk to you about one of the most important things we just discovered. It's published in May, of a new form of a pathophysiology axis that really explains a lot of things in MMA. It's called post-translational modifications, and there's a very unusual one that's novel and new and formed. Dr. Head discovered this. Dr. Buckley was mentioning his talk. I don't have time to go through it today, but it's very important. So overall, these are sort of like our summary statistics. We've seen about 1,250 patient visits. Many of you in the audience have seen us. We've created about more than 100,000 biospecimens from all the blood samples and urine and everything you've all donated to us. We've generated more than 20 animal models. You need these to do test therapies. We've written hundreds of papers on MMA and PA, and we've produced intellectual property that people are interested in. And in fact, I'm proud of this too. We help everybody, and we've helped enable 3 new treatments for MMA that are going to go into the clinic: mRNA therapy, genome editing, and AAV, conventional AAV. And then for those who have PA, you're next in line. And we have a different mechanism for this, and it's definitely going to go. It's with the government, and I'll tell you a bit about that later on. So I'm not going to go through much about the treatment approaches. I think you heard some of the things. I've listed some of them. If you're a B12 responder, you're very lucky. Most patients don't respond. But if you are going to be have a much, it's really night and day in terms of non-responders versus responders in terms of how the clinical outcomes are. These are the things that don't really work. That's almost everything on the list, except for B12. So then you have to step back and say, wait a second, why doesn't conventional management work? This is where it's going to get a little theoretical, okay? Just get ready for this. There's a number of reasons. I'm just going to give you part 1, 2, and 3, okay? The first one is related to metabolism. This is a harder one to grasp, but it was mentioned by another speaker, so this is fair territory for me to go. as you try to metabolize amino acids, this is what happens in your body. You try to get rid of the nitrogen group. That's called transamination. And you make something called a keto acid. That's the second step. And then once the next step happens, that's the branched-chain keto acid dehydrogenase step. That's irreversible. All of a sudden, once you do that, you can't go backwards. And you're stuck. Then you have to metabolize. And this is a fundamental problem. You're stuck with something called obligative oxidation. So that's one thing to think about. There's another thing to think about, and that's this. This is, again, a depiction of the pathway. It's a little complicated. But if you have a block, you build shunt metabolites and toxic metabolites inside your cells. MMA and methyl citrate are two of them. And then there's secondary effects on the mitochondria and oxidant responses of the cells. And then there's this. This is the endogenous production of metabolites. So cells that don't have full correction are going to keep producing the metabolites. And this makes it very difficult for canonical biomarkers. So if you just go, hey, I'm going to measure someone's MMA level, let's make an example, and someone has a liver transplant and you measure their serum MMA in a clinic, say, for example, the reading you will get back looks just like a patient who doesn't have a liver transplant because the MMA is being made other places, like the skeletal muscle. Okay? So this tells us a bit about the disease and the difficulty of biomarkers, a theoretical construct will you have. Then there's yet another reason why, and this is the really sort of, now we're going out to the top level of like, this is the most difficult thing to understand. And this is this new aberrant post-translational modification called methylmalonylation, which Dr. Head in my group discovered and really studied. This is an amazing, amazing paper. Get ready. This will take you a day to read. It's absolutely amazing. But it got a lot of attention, and Dr. Vockley wrote a special article to talk about how important this is, and he talked about it in his talk, why this is a new way to think of the pathophys. I'm not going to go through it. I don't have enough time to go through it with everybody. But I'm glad to talk to you about it if you're interested. The next newsletter, yeah, we can. So now I'm going to switch over. That's sort of a bit about background. And we're going to switch into the next sort of final chapter. So for those who like drink your coffee, get ready here. We're getting close to talk about new therapies. And these are the ones that are genomic-based therapies. It's RNA editing and gene therapy. I don't have time to go through them all. We can't. There's too much material. I'll make a few comments that might be of interest at the end. But I'm going to switch to talk about biomarkers. We keep hearing about biomarkers. What is a biomarker? Okay? This is what it is. You can read the definition. And what's very important about it is the last thing. This is why there's a real intense interest in this. You can monitor the patient, but perhaps it can be used as a clinical outcome marker. That's a huge hurdle to overcome. But if you get that, that is a massively important thing for enabling a new therapy. Because then you could just maybe make a blood or some other type of a measurement for a patient, and then you would have a correlation with outcome. So if something did something very important to these biomarkers, that could be something a person could use to the FDA and say there's clinical efficacy. Okay? Super important. But there are none for MMA and PA. And we have significant developments, not just me, a lot of people in this area. But there's no FDA-approved biomarkers. And by the way, I have to thank Dr. Shlachkov. I stole his slides. They're beautiful, and I'm using them. But the diagnostic markers that are obvious are, well, what about MMA level, right? What about propionylcarnitine? C3, what about methyl citrate? Those are real good ones. And we measure that in our patients, right? Let's use those. Well, it doesn't work. This is from Dr. Manoli's paper. Why don't you point out, this is log scale. So instead of going 1 to 10, it goes 1 to 100, it's compressed. And what you can see here is tremendous variation in MMA levels in patients that are being measured serially in the hospital when they're under control. It's variable. So use this as an analogy. Say you're trying to go on a diet, okay? And you weigh 100 pounds. Let's make pretend you weigh 100 pounds. You're going to weigh yourself. So one day you weigh yourself, and all of a sudden you weigh like 100. And then you go on a diet for a day, and you're like, well, maybe it worked. Let me weigh myself. And you weigh 110. And you're like, whoa, wait, this is impossible. And then you weigh yourself the next day, and you weigh 90. That scale is not any good, right? This is the same idea. This is what this is telling us. It's not a reliable way to look at probably an outcome by using a static metabolite measurement. What about methyl citrate? This is from Dr. Shlachkov's work. Well, it doesn't tell us the difference between someone who had a liver transplant with PA and someone who didn't. It's not any good. You can't use it. Same with C3. Now, the problem gets even further accentuated when you try to think about doing something like gene therapy. And I'm showing you, this is an example I'm going to show you again. This is a famous mouse. He's been published in the paper. So this is all, real approved by the journals and everything. That's an MMA mouse up in the upper right, or upper left. Very sick mouse. And that's how, that's a really good looking mouse because it's alive. Most of them die in the first day or 2 days of life. And when you do gene therapy on these mice, a single dose of an AAV, lots of different ones we've tested, they end up looking like this. You can't tell the difference. They're absolutely perfectly normal looking mice running around their cages. They live a long, long time. We said, aha, they're going to have 0 MMA level, right? Wrong. Their MMA levels are they're hundreds of fold elevated because it's not predictive, right? So what's predictive? Well, that's when you have to go back and say, we have to develop better ways to look at this. And here's one of the ways we use. And this is a way. There's other ways. Using isotopes. And this is a stable isotope, single bolus, oral breath test method. In mice, we inject them, but in humans, you can oral. And you give this non-radioactive heavy carbon. And what happens in this setting, and this is unusual, this doesn't always happen for all labels, very quickly it's taken up by the liver and converted to its active metabolites, so to speak. And if a person or a mouse has the enzyme activity, that's the triangle, mutase in their liver, what's going to happen is that heavy carbon is going to be released into the breath of the mouse. Then you have to catch the breath of the mouse and say, how much heavy carbon, Mr. Mouse or Mrs. Mouse, are you producing? And that's not easy. That requires invention, devices, special methodology. That's how you do that down below. It turns out you can do the same thing in patients. Here's some of our patients that are undergoing a similar type of a study. And in patients, it's non-invasive. There's no injections. It's just drinking or getting G-tube on a label. Now back to our mice that look so good, except that super high MMA. Well, guess what? They oxidize. That's why they're doing so great. They can metabolize the label. So, in an extension, you can do this in patients with MMA. And I want to thank all the patients who participated in this study. Again, a famous paper published by Dr. Manoli showing that this can be used in patients with MMA. And if you look at the curve here, some of our patients have liver kidney transplants, very high MMA levels. But on this test, they look just like a healthy 20-something-year-old control because they have the enzyme activity in their liver. The same thing can be observed in some patients with propionic acidemia versus patients who are not transplanted below. You see? I don't have a pointer, I'm sorry. It doesn't work on the screen, I can't figure it out. So if you just see the bottom curve, how there's a lot of variation. That's because there's a lot of variation in patients. Some metabolize a little more than others. It's very reproducible and repeatable. We studied that. And that's published, by the way, if anyone wants to look that up. You can even take it another step, and this is an absolutely unbelievable paper that Dr. Shlohachkov wrote. It's so sophisticated. I can't really do it justice, but I'm just going to tell you, if you see the title, how sophisticated it is. This uses a combination of machine learning and special statistics to figure out from hundreds and hundreds of measurements, having a computer just say, what's the most important thing, and permuting it. And to do this, we needed supercomputers to run the programs. And you can sort of ask, Oleg can comment when he gives a talk on this. What is all this mathematical computational manipulations tell us? Well, they identify a few very interesting biomarkers that I don't think we would identify if we just tried to look at them. You wouldn't. You need a supercomputer for this. One of them was propionate oxidation. And in PA, it really correlates. It correlates with a lot of outcomes. So it's correlating. It doesn't prove it, causation, but it correlates. So this is on your first step to having a biomarker that could be representative of a clinical outcome. And it makes sense because what you're doing here is looking at how much enzyme activity someone has in their liver. You're not just measuring a level. It's an active metabolic process. And it correlates with things like other metabolites, IQ, and and height. So these are sort of really important clinical endpoints, right? All right. That's just a touch on biomarkers. I'm going to come back. There's even more about biomarkers I'm going to say next. Next, I'm going to sort of go on a tour of AAV gene therapy, and it's going to be a quick tour. And it might require some discussion with people afterwards. because I'm not really going to go through a lot about what AAV gene therapy is, other than to say AAV stands for adeno-associated virus, and it's been reconfigured over the years to remove all the elements of the virus in itself. By the way, it's what's called a dependovirus. It doesn't replicate on its own. It needs a helper virus to replicate. And then everything's removed from the AAV except for the very ends called the inverted terminal repeats. And then your gene of interest is put inside of it, and then you can package them into different flavors. Like imagine, there's different strains of the flu, right? Every year you get a different flu vaccine. You can do the same types of things with AAV, and you can change the shell on it, the outer coating of it. It makes a very different, the properties of it change. So there's a lot to say about that. I'm not really going to go into it, but just to say that there's standard ways to do this. And so we did this. We did this over the years. Tremendously effective. In fact, the most effective treatment in the mice, nothing comes close to it, because it can always rescue the sickest mice quickly. AAV systemic gene therapy nothing else can do that. And again, here's our famous mouse, and that's what it looks like. It's not, this is a therapeutic approach. There's other ones. And by the way, other treatments that use editing and mRNA therapy also work in mice. They don't work in these knockout mice, though. So there's a fundamental problem, too, and I want to just say this. Little tiny baby mice don't always fully recapitulate the human. A little baby mouse is about the size of a jelly bean, for those who eat jelly beans. And they weigh about a gram, 1.4, depending on the strain. So they're also equivalent to a premature baby, so someone that's very, very premature. So there's a lot of, theoretically, to say, I treated a baby mouse. It's not the same as treating a full-term infant, for example. So you have to keep that in the back of your mind. So this constantly motivates the search and the development of better new mouse models. And I'm going to tell you about one that we made for MMA, okay? And the way you do this, and this is going to be complicated, is you make a rescue gene that you put into the germ line of the mouse. And this rescue gene is going to cause the expression of the missing enzyme only in the skeletal muscle. And then you breed that to a knockout MMA mouse. And so what happens is that mouse is going to have that only enzyme in its body is going to be coming from its skeletal muscle, which is not normal. That's not normal physiology. And you ask the question, what happens to the mouse? Well, the first thing you find out, if you look at this curve over here, and boy, I wish I could get the pointer to work on this. The mouse doesn't seem like it works. If you have to look over here, that little slotted line, that's what happens when the mice don't have the transgene. 100% die right away. When they live, they can actually live quite some time. And then they start dying off because they're not fully corrected. They have enzyme only in their muscle. The liver doesn't have any enzyme. This is key. 0. And if you look at these mice and if you put them on a diet that causes its brother or sister mouse to become morbidly obese, that's almost the size of a small rat, these mice are eating everything. They're runted. They don't grow. Why is that? This is part of methylmalonic acidemia without proper treatment. There's a growth problem. The mice recapitulate this. And you can rescue that with different types of therapies. The other thing you can use these mice for is to discover biomarkers that are coming from the liver. And these mice produce something called FGF21, which is very interesting in MMA. It's massively elevated in the mice. And when they get a new liver, for other reasons, it goes down, just like patients. Patients have the exact same finding. Before a liver transplant, they're super high. After a liver transplant, they go to normal. So this is a dynamic biomarker. So that would be the question is, can we use it in gene therapy? Answer, yes. So I'm not going to go through all the experiments we've done. We've done a number of these with people over the years. The ones I'm going to mention quickly we've done in partnership with Selecta Biosciences and our colleagues at Harvard, Luc Vandenberg. And I'm just going to show you an example of sort of what you can do with these new models. And what you can do is you can make a viral vector, a gene therapy vector, to deliver the mutase gene only to the liver. That's really where you need to correct. Always, you must target the liver in these applications. It's the most important site of metabolism. And you can inject the mice, and you can ask the question, if you look at the first graph over here. Boy, I wish I had some way to point. But you can see here the graph that has MMA level. So MMA goes way down, as you would expect. Now, don't forget, in mice are in a cage. They're eating the only same diet. They don't go out and have an a la carte lifestyle. They're in a cage eating a certain type of chow, sitting in a cage, okay? So diet control is very different in mice than in patients, in humans. But the other thing of note is the biomarker, this FGF21 marker, really responds beautifully, just like the MMA does. So it's dynamic. Now, the other thing you can do with mice is you can take those same mice and you can ask the question, and what I'm showing you in this graph with the little dots, is can they oxidize propionic acid? In other words, I remember I was telling you before about the biomarkers. The answer is yes. And if you'd see there, they can do it after 12 days, and it lasts for 1 year. That's the equivalent of 30 years in a human. It's durable. And if you take their livers out, that's what the next panel shows. That's the enzyme. It's still in their liver 1 year after the mice were treated. So it's durable. So this is what's really great about this mouse model, and a lot of people have used it. All the people that have enabled treatments for MMA have used this mouse model because of the reasons I just articulated. So the next thing that can be done with these mice, and then as a therapeutic approach, is it can be combined with an agent to modulate the immune system. In this case, it's called mTOR, or Cell 110. This is developed by Selecta Biosciences, and they're here today. And this is a encapsulated particle that's degradable, and inside of it, it contains an immunomodulatory compound. So the idea is to give this, and it will produce an effect on the immune system, probably to mitigate antibody formation. That's one of the things for certain it does. And then could be T cell effects. So this is something AAVs can have a problem with immunity. So this is a real important advance to be able to combine this with AAV. In fact, that was done. And there's even a saltatory benefit of this, which is it seems to make the AAV a little more potent in the mice. So you might have to give less of it to get more for your money, so to speak. And that's been published if you want to look up that paper. So with a lot of other experiments that were done, and I'm not going to go through them, many were done by Selecta Biosciences, there is yet another new therapy for MMA that is going to be coming to the clinics. And this is from the OAA newsletter. That's why I took the title. And that's what it is. It's a depiction of what's going to be in this new treatment. It's going to have this immune modulatory particle and an AAV to do liver-directed gene therapy. And I have pictures of the people from Selecta Biosciences there at the meeting over there. And if people have questions, they can talk to them. I think there's other colleagues from other companies here. I saw Dr. Carrillo, she's from Moderna. Dr. Guskin's over there from Logic Bio. So that some of the companies are here, and I think they'd be excited to talk to anybody who wants to talk to them. So if we looked at a summary table of sort of what are the newest things, I've listed them here. There's RNA therapy, there's gene editing, and then there's this AAV canonical plus the immune modulation. They're all going to be IV. The RNA therapy eventually will definitely require multiple IV infusions. Everything else is one-time infusion. You can see the way they work. Those are the mechanism of action. And I put on side effects. These are possible, probable. We don't know. This is experimental. This is what you do in a phase 1-2 trial. You have to check for side effects. And any medicine that's new will have side effects. For certain, they will. And there's some that are AAV related and some that could be lipid particle related. We don't know. We're going to find out. You have to always think about the risk benefit here, though. If someone, if this is an alternative to a liver transplant, would someone consider it? Or is it going to improve the quality of life? These are the individual decisions that each patient that wants to do this will have to take up with their care providers and their families. But this is sort of just a boiled down summary slide. It's just for the audience. I'm glad to discuss later. So, can we use the same approach for PA? Yes. Does it work? Yes. Increases survival, increases the metabolic status of the mice, allows transgene expression in the liver, and oxidizes. We published this in 2011. And recently, we've partnered with NCATS, the National Center for Advancing Translation Science, to develop this initiative called PAVE-GT. You can read about it. That's the website. And we were able to nominate a few disorders. One's propionic acidemia A type and MMA B type. This is going to happen. And we developed new mouse models that are much better than anyone made with PA mice. There's a serious problem with those models I don't want to get into. I'll talk to people afterwards. But here's our lead vector. You can see what you're seeing here is the mice that have PA that don't get the treatment are sort of on the black and red. They die right away. That's what happens to a severe patient, unfortunately, with propionic acidemia. If they're not treated right away, it's lethal. The mice, if you want to use a model that shows a clinical effect, you have to recapitulate the clinical syndrome that's in the patients, and that's lethality. Lots of studies on PA use mice that don't have lethality, so it's hard to say how does it accurately recapitulate the clinical syndrome? The answer is probably no. And you can see here, there's a dose response. So lower doses don't work. You have to keep going up on the dose till you find a dose that works. So this is really excellent. I don't have time to really go through this. It's super exciting. Dr. Lee is here. Dr. Lena Lee, she's there. She's been leading this effort. She could talk to you about it. It's super exciting. And we have an orphan drug designation by the FDA for this, awarded to NCATs. So this is moving to the pipeline. There will be gene therapy for PA, PCCA type, and we're working on it for B type also. How about cobalamin C? Can it work? Yes, it can. It's the same idea. Make the mice that have the real disease. They're very sick mice. And Jen and Pauline is in the back have been working on these mice. They really have this human syndrome. They're growth retarded. And we've done things to the mice, like to say, okay, well, let's try to do the treatment you do on a patient, right? Inject your child every day with high doses of cobalamin. It's not, no one wants to do that. Or you give them a single injection of AAV. What happens is if you just have the mice without any treatment, you can see the mice, that's the red, 100% die. The mice that are the littermates that receive cobalamin, they will live and they're affected. Some die. If you use AAV gene therapy, just AAV gene therapy, it's quite effective. That's the green line. If you combine them, the next one is yet a different form of gene therapy that is a special cassette, a special vector that is super potent. The mice basically live. You can see how long they live, 100-something days. That's pretty impressive for a mouse. Remember, 3 years, or 1 year in a mouse, 30 years in a human. Oh, yeah, okay. So it works. Now I have to say this about the CBLC mice, because this is the first thing everybody wants to know. Do they have the eye problem? Answer, no. Mice don't have maculas. We spent a lot of time trying to study this. So it doesn't mean this isn't very useful, because we could use such mice and do gene therapy studies on their eyes and show gene delivery and transduction and all those sorts of things you want to do. But unlike the MMA and PA mice, which have a real careful recapitulation of the human phenotype, these mice don't have that. And again, it's part of the difference in physiology between mice and humans. They don't have a macula. Macula is a specific area of the eye that's affected in the children, many of the children with CBLC. It causes a serious problem, vision loss, when it's damaged. So to conclude here, I want to hopefully I've tried to impart to everybody and explain how there are potent AAV gene therapies that exist, that many in the lab, we have many, we've had them for more than a decade in some cases. They're moving to the clinic for MMA. PA is behind, but it's getting there, and then CBLC is in progress. We're looking for partners for CBLC, so there are companies here that are very interested. Please come talk to me. Somewhere in the clinic. The other thing I wanted to emphasize to the group, and I hope I tried to explain this, is that the testing of these things in animal models is really, really important to enable the treatment and get an idea about how potent it is. That's super important, and it's informed by clinical research. That's the other thing that's really important to remember. You need both. And that benchmarking therapies can really help. And this is what we do in the animal studies all the time. What's the best dose? What's the best vector? What's the best capsid? So there's ways you can combine those things, and in the real world, people tend to not want to test one class of therapy versus another. In the research world, we do that every day. And it's important to help get an idea of how effective it is, the relative potency. And to say that it's really great to be here, especially with all the different companies and trials getting involved, this is really, really important. We need partners to help facilitate translation. We're mainly doing research and clinical research and laboratory research. We need companies to come in and be very interested and partner with us to get these things to the clinic for the patients. That's very important. And I applaud all the companies getting into the space. So with that, I will close and see if anyone has any questions for me. I think we had a little snafu there with the audio visual. But if anyone wants to ask me a question, please. I'd love to have a dialogue about it.
Thank you, Dr. Venditti. I'm going to open it up. Does anybody have any questions? Oh, sorry.
So I hear you saying that PA is behind in terms of gene therapy. Could you give some sort of timeline or estimate?
We have an ODD. We would like to file an IND. For those who don't know, ODD stands for Orphan Drug Designation. What really matters is the IND. That's the Investigational New Drug Application and Approval. We're on the way to that. I can't really say anything more about the timeline other than it's going down. And one of the things, again, for the practical side, for those trying to develop these new therapies, making AAV is very, very expensive. And we have someone that is funding this, guaranteed. Indirectly, it's the United States government, the taxpayers, you all. It's coming from the National Center for Advancing Translational Sciences. So funding has been secured. Manufacturing is proceeding. And the next steps are, it's harder to predict exact timelines, but I think Oleg is the lead clinical investigator on this with myself for PCCA. So I don't know. I think we would love to be able to dose someone in about 2 years.
Okay, so I have a follow-up question. Mm-hmm. Since we obviously have multiple other trials going on right now, would participation in any of those 3 trials preclude someone from participating in a gene therapy trial?
We're not there yet, but we would probably, if someone was on a small molecule therapy, for example, I'm not saying anything about what it's going to happen because that's the future and we don't know what the future is. Then you would want to do something like, well, how long does that medicine take to wash out of someone's system? And you would probably say something like that. Like say it washed out in a month, then maybe you'd say 2 months or 3 months. That would be for some sort of small molecule. If someone's on a different type of gene therapy for PA, for say someone developed another one, then the answer would be no, they're not, that patient wouldn't qualify. If they already got a gene therapy for PA, you wouldn't take them in and do another one. So that would be probably a disqualification. Now, AAV-based therapies, and I must say this to the people in the audience, it didn't come across as clearly as I wish it did. People make antibodies against different types of AAVs. AAV, adeno-associated virus. In this room, in the adult population, right here in this room, probably 75% of us will have antibodies against AAV serotype 2, for example. We're not using that in our application. But if someone has an antibody, they're not going to be able to qualify for the trial. Because the reason is because that will block the effect of the AAV, the therapeutic agent in the patient. So that is going to be a disqualifier, and that has nothing to do per se with whether they were on another trial. It would depend on what the trial was. Something that gives back a gene, let me just give you an example, again, from another field. Right? We're getting technical here, so just bear with me for a second. There have been studies in other fields where people do enzyme replacement therapy, ERT, for lysosomal storage disorders. It's a different class of disorder. It's not MMA or PA. But as an example, if someone gets that enzyme and they're immune naive to the enzyme, they can make an antibody response to the enzyme. So if someone got, for example, I'm not saying this is going to happen, but if someone got such a therapy that produced more enzyme, for example, from a lipid nanoparticle, and all of a sudden they didn't want to get that anymore and switch to gene therapy, that patient could make an immune response to the gene therapy vector because they're getting an immune response to what's in the particle because their body doesn't have it. That's always going to happen. So in such cases, it would be specialized testing on each patient to determine, would you qualify? There will be patients who would not have such a reaction. They would qualify. You would just do a washout. That's how I think we should do it. Now, companies usually don't do this. They don't want you to come into a trial if you ever got anything else in another trial. And I'm speaking that on behalf of my corporate partners, but they could take the floor in a minute and see what their opinion. I'd be interested to hear their opinions on this. So I don't know if that really fully answers your question. It's a discussion point, but those are some things we would consider. We would really be, we don't want to say no to anybody, put it that way.
Does anybody have any more questions?
I'm guessing this won't help the older kids like Michael, gene therapy.
It brings up a bigger discussion, but here's something just to consider, right? If a person has hyperhomocystinemia, chronic, long-standing, it's probably causes other effects on the body. So and again, the hyperhomocystinemia syndrome, it's a different discussion for the MMA PA audience. Homocysteine itself probably has certain effects on cells. It could be partially toxic in some ways. So if someone could get a systemic gene therapy, for example, and have much lower levels and then not need injections, it might not be a curative therapy if they have a neurocognitive impairment. It might not cure that. It probably definitely will not cure that. But it might make the quality of life better for that person if they don't need to get injected, they don't need to take betaine, and they don't have to worry about hypercoagulation associated phenomenon due to the high homocysteine and low methionine that some of the patients have problems with. Now, that is not the reason you would do a gene therapy right away in someone with CBLC. But for example, that could be a benefit in somebody eventually if it was first tested in patients where there's a real need-based indication before an adult would receive it. But it would be a debatable topic because someone might say, if someone has hypercoagulability, you should mitigate that risk. We know that it is associated with strokes and heart attacks and clots and DVTs, all sorts of things that are not good.
Yeah.
What's going to happen in adults with CBLC? We don't know. But we do know for certain that patients that have CBLC that are undertreated have increased risks of coagulation problems. It's absolutely irrefutable. So I don't know if that answers your question.
Yeah, well.
Sort of.
It'll be longer than the 5 years.
I know.
Yeah, it'll be.
Reverse brain damage and keep praying.
Yeah.
Well,
Does that have
I
anybody?
wish we could say something else, but to be realistic about it, it's not going to, it won't do that. These therapies won't do that.
Yes, thank you for that nice speech. My question is, I know you guys have a cure for MMA with people without liver transplants, and I know you guys are currently working on the cure for people that already had liver transplants, but do you guys have an estimated date of when that cure for people who have already had liver transplants might be, like an estimated time for the cure?
Right. So some of the things I talked about, if you had a liver transplant, you're not going to be a candidate for any of them for these gene therapies. Okay. But other people are working on other things that, like Dr. Vockley, if you were here earlier, there's other small molecules that people are working on and different types of classes of things that could be useful. And I should also tell you, as an aside, I didn't go through this, but one of the students in our lab developed a central nervous system specific type of AAV gene therapy that in mice, when it's given by vein, goes into their brains. And one of the things we wanted to do in that paper was to consider, and this is theoretical, it only works in mice, whether if we had something like that, maybe patients would be considerate if they had MMA or PA because it could protect their brains by giving the gene back inside the brain without having to inject anything into a person's brain. You just get an IV. So it's pretty, I didn't go over it because it's sort of a little esoteric. But in mice, we have a publication on this. I'm glad to send it to you. It definitely works. So.
Any other questions for Dr. Venditti? All right. Thank you so much, Dr. Venditti.
Thank you, everybody.